Hello everybody, my name is Cristiano Jayme. I'm from Brazil, University of São Paulo, campus Ribeirão Preto. Ribeirão Preto is known as a city of cold beer. It is a great pleasure to talk to you, and today I will present the work intituled Preparation, Characterization, and In Vitro Evaluation of DNA-based polymeric film for tissue engineering. The use of desoxyribonucleic acid (DNA) as a functional biomaterial for therapeutic, diagnostic, and drug delivery has been prominent in the recent years, but the use of this polymeric film as a generation biomaterial is still limited. So the end of this study was to evaluate the biocompatibility and the interaction of this DNA-based polymeric film with primary human fibroblast, application for regeneration medicine, and wound healing purpose. The tissue engineer is an interdisciplinary field that involves technology from material science and cellular biology. It's covered to develop an application of a biocompatible and biodegradable material that act as a matrix from cell growth and a drug delivery called scaffold. In other words, the tissue engineer is a field that has received a lot of attention because he has the ability to regenerate damaged tissues throughout the cells, structure, and bioactivity molecules. Scaffold acts as a support which the cells are grow to stimulate the tissue in vitro. The scaffold development and manufacturing project are important areas of biomaterial research and also within tissue engineering and regenerative medicine. Scaffolds are defined as a three-dimensional solid. The solid can be a polymer, natural or synthetic, ceramics, and the others. 3D biomaterial designed to transport body liquids and gases and promote the cell interaction, improve cell or tissue, and create a micro ambient to promote the extracellular matrix and mimic the ambient. So the first step is cell isolation of the patient and the cell is patient in the in vitro environment. And with the proliferation and the growth of these cells extracted from the patient, we interact these cells with a scaffold. We have many ways to build this scaffold, but currently the 3D printing has gained a lot of promise. Because of the precision of the 3D printing process, we can build a custom structure. After the scaffold are built, the cells extracted from the patient are placed in interaction with the scaffold and with specific growth factors for the formation of the tissue of interest. At the end of the interaction process and with the formation of mature extracellular matrix, the scaffolds with the cells can be replaced in the patient and assist the tissue regeneration process. So this is the basic cycle of the tissue engineer. Started with the extracting of the cells from patient, building the scaffold, interaction the cells with this scaffold and the growth factors, and the reimplant this scaffold with the cells into the damaged tissue. The desoxyribonucleic acid DNA is one of the most important biopolymer present in living organisms. In addition to carbohydrates and proteins. The DNA molecule is composed of desoxyribose sugar and phosphate molecules, and four nucleic acids. In the last 2 decades, the DNA has used as a functional biomaterial and has been used for numbers of applications. These applications prove its potential as an active component in chemistry, physics, and computer science, medicine, nanotechnology, and the others. This great attention is in great part because the DNA have ability to self-assemble in addition to the low cost of synthesis and a high biocompatibility. In the case of regenerative medicine, has been also prominent in the recent year, but much of this focus is mainly on develop DNA nanostructure and the drug delivery system. Some studies conducted by our group have demonstrated the high biocompatibility and the excellent control of drug release properties of DNA-based polymeric film. However, no reports have been found in the construction of this DNA polymeric film has a growth cells support based in DNA for wound healing and helping in skin regeneration, focus on tissue regeneration. The use of the high biocompatibility polymers, such as DNA, may offer a promising strategy to build a new and more complex biomaterial for tissue repair and application in the future. So in this study, we focus on understanding and evaluate the biocompatibility of DNA polymeric-based film and a primary human fibroblast. The polymeric film has prepared by a solvent casting method. This method involves dispersing a film formation solution on a support, usually a petri disc or glass, followed by the slow deshydration, leading to well-structured 3D material. Then the morphological structure of the DNA polymeric film has observed by SEN, scanning electron microscopy, elementary microanalysis, and atomic force microscopy. The interaction of the polymeric film with the human fibroblast cells was evaluated by classical MTT assay and rosazurine colorimetric assay, cell cycle kinetics, cell viability, and oxidation stress by flow cytometry. In addition, cell morphology and adhesion were evaluated by immunofluorescence and migration assays. The first step of our work is the morphological analysis of the DNA-based polymer film, and this figure 1A and 1B show the morphology profile of the DNA polymer film evaluated by SEN and IFM respectively. The SEN image show a flat morphology of the DNA-based polymer film to approximately 200 micrometer. It is still possible observation the presence of the elements like carbon, oxygen, sodium, and phosphate throughout the analysis of elementary composition obtained by elementary microanalysis, which are chemical elements of the basic composition of the DNA molecules. And the FM results presented consist with the same analysis in different approaches, since it also has a high flat surface can be observed. The second step of our work, we analyze the cell adhesion and the spanning process with the cells under the DNA polymer films. The cells of human fibroblast sits on the surface of the neopolymer film has evaluated up to 72 hours of incubation, and it was observed that the cells were able to grow and forming colons and show a expanding independent of the neopolymer film concentration. Our work, we varied the DNA concentration in the construction of DNA-based polymer film. The adhesion test carried out also indicates a time of 48 hours for a better adhesion of the cells under the DNA polymer film. This data is confirmed, observed by micrographics. Almost no attachment was observed on 1 and 2 grams per liter in the construction of the polymeric film. Only few cells attachment to the 0.5 grams per liter DNA concentration after 24 hours of seeding. However, 72 hours post-seeding, the cells were more proliferate in 0.5 concentration and the neopolymer film compared to the high concentration of the neofilm 2. And the cells have a very similar shape compared to the control. The SEN analysis confirmed this shape of the cells compared with the control. And to confirm this cell adhesion process, we do the confocal analysis and target the neopolymer film with CD31, and the cells, nucleus cells, are targeted by DAPI in blue. We need mark the DNA with CD31 because the DNA don't show apparent autofluorescence. The cells can attachment on the polymeric film and proliferation. But we need now evaluate if this polymeric film has toxicity for the cells. And the results of cell viability are assigned by MTT, resveratrol, and via count SI. 48 hours after the seeding process of the cells in contact with the DNA polymer film. In our analysis, the high cell viability was observed in all the 3 independent experiments, and no significant activity different was observed between control and the cell seed on the DNA polymer film groups. So, independent of the DNA concentration, which confirms the high biocompatibility of this DNA polymer film with the cells. We monitored the intracellular reactive oxygen species. ROS formation was monitored 48 hours after seedings of the cells on the DNA polymer film at different concentration of DNA. This is measured by the conversion of a non-fluorescent DCFDA to fluorescent DCF using a flow cytometer and a microplate reader. As indicated in the figure 4A, a very slight redox imbalance was observed between the cell seed on the DNA polymer film, the blue line, and the control, the red line groups. Once both groups showed the similar green fluorescent signal, the relative fluorescent unity was quantified by a microplate reader for different concentration of DNA, and different was observed between the low concentration of DNA groups and the control groups. This information means to us that DNA in this concentration was able to shield the oxidative stress during the cell proliferation. And in your case, in your study, we can think this is the better condition to apply. I forgot to say in the previous slide, we analyzed the key I 67 and the cell cycle proliferation of the cells under the DNA polymer film. And these studies indicate to us the cell under the polymer films has a more Ka 67 compared with the control. The Ka was used to see the cell in the proliferation process, and some form the DNA can accelerate the proliferation of the cells under the polymer film if compared with the control cells. And the cell cycle analysis compared the cell seed in the polymer film with the control showed to us the we not have a significant difference in the phase of the cell cycle in the 72 hours if we compare the cells under the polymer films and the controls. And in this figure we can show the migrations I, and we can see no significant difference was observed in cell migration between control and the cell seed in the DNA polymer film groups in the first few hours of incubation. Whatever, the cell seeds on the DNA polymer film show a significant high migration rates after 21 hours than the control groups. And after of 16 hours, the plate were observed hour by hour to close the scratching area. The cell seed in the DNA polymer film took about 61 hours to complete close the scratching area, and the control took about 16 hours to close the sketching area. And in this case, we can conclude that the polymer film can help the cells to close the scratching and facilitate the migration of the cells under the surface. The results and the present study suggest that the neopolymer film base has a new biomaterial for wound healing and regeneration medicine, and describe the growth behavior of the dermal human fibroblast seed on it. The proposed method allowed to obtain a polymer film with a sustainable surface and topography characteristics to support the cell adhesion process and the proliferation these cells under the polymer film. The low cytotoxicity of the DNA polymer films indicate the the convergent with the literature and confirm it is higher compatible material. I would say thank you very much for the opportunity and your attention, and congratulations the committee, the organizing committee, for this great event. And here are my contacts in case of any doubts. Thank you very much.